Welcome to Biotanemy.
Yeast-Two-Hybrid System. The Yeast-Two-Hybrid System, or Y2H, is a technique used to identify the genes that encode proteins that are physically associated with a given protein, with the help of yeast cells. The Y2H system is used to study protein-protein interactions, DNA interactions, or protein-DNA interactions inside the cells. The Y2H system consists of bait protein, prey protein, and a reporter gene. A bait protein contains the protein that we are interested in studying. For example, PGBKT7. It contains a Gal4 promoter that drives the expression of the gene of interest in yeast. It is induced by galactose and repressed by glucose. It contains a Gal4 DNA binding domain, which was fused with our bait protein. The bait protein used two reporter genes, ADE2 and HIS3, the genes that are involved in adenine and histidine synthesis, respectively. Also contains multiple cloning sites and selectable markers, like ampicillin resistance gene. And finally, an origin replication to propagate them in E. coli cells. They don't have ARS to replicate in yeast cells but have a TSUN sequence. The TSUN sequence allows the plasmid to replicate in between the daughter yeast cells and ensure their stable maintenance in the cells during replication.
Prey protein. The gene that encodes for prey protein is cloned into a vector which will fuse it with the Gal4 activation domain, which will help them to activate the reporter gene when it interacts with binding domain, which is present in the bait vector. Example: PGADT7. They have a Gal4 activation domain, autonomous replicating sequence, the LEU2 gene as a selectable marker, and an origin of replication to allow them to replicate in E. coli, along with a selectable marker, multiple cloning sites, and a terminator sequence. After the construction of bait protein and prey protein, the plasmids are then transformed into the same yeast cell. The cells are then grown in the selective media. If the BD and AD will interact with each other in the yeast cells, they will grow in the selective media. Hence, the growth indicates that the bait and prey protein has interacted with each other. Applications of the Y2H system are: 1. Mapping of protein-protein interaction. 2. Functional validation. 3. Screening for new protein. The limitations are: 1. False positives or negatives. Non-specific interactions may occur in the yeast environment at different cellular context. 2. Complexity of interactions. Some protein or DNA interactions require additional cofactors or specific cellular conditions, which may not be present in the yeast cells. That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.